Hi everybody, I'm John Capello. You're tuned into the Top Wrench channel, and this is a narration of Principles of Anatomy and Physiology, 12th edition. I'm on chapter 3, and this is cellular diversity. Cellular diversity. The body of an average human adult is composed of nearly 100 trillion cells. All of these cells can be classified into about 200 different cell types. Cells vary considerably in size. The size of cells are measured in units called micrometers. 1 micrometer, abbreviated as is equal to 1/1,000,000th of a meter, or 10 to the negative 6 m, or 1/25,000th of an inch. High-powered microscopes are needed to see the smallest cells of the body. The largest cells, a single oocyte, has a diameter of about 140 micrometers, and is barely visible to an unaided eye. A red blood cell has a diameter of 8 micrometers. To better visualize this, an average hair from the top of your head is approximately 100 micrometers in diameter. The shapes of cells also vary considerably. They may be round, oval, flat, cube-shaped, column-shaped, elongated, star-shaped, cylindrical, or disc-shaped. A cell's shape is related to its function in the body. For example, a sperm cell has a long whip-like tail, a flagellum, that it uses for locomotion. The disc shape of a red blood cell gives it a large surface area that enhances its ability to pass oxygen to other cells. The long spindle shape of a relaxed smooth muscle cell shortens as it contracts. This change in shape allows groups of smooth muscle cells to narrow or widen the passage for blood flowing through the blood vessels. In this way, they regulate blood flow through various tissues. Recall that some cells contain microvilli, which generally increase their surface area. Microvilli are common in the epithelial cells that line the small intestines, where large surface area speeds the absorption of digested foods. Nerve cells have long extensions that permit them to conduct nerve impulses over great distances. As you will see in the following chapters, cellular diversity also permits organization of cells into more complex tissues and organs. Aging in cells. Aging is a normal process accompanied by progressive alterations of the body's homeostatic adaptive response. It produces observable changes in structure and function and increases vulnerability to environmental stress and disease. The specialized branch of medicine that deals with the medical problems of care of elderly persons is geriatrics. Gerontology is the scientific study of the process and problems associated with aging. Although many millions of new cells normally are produced each minute, several kinds of cells in the body, skeletal muscle cells and nerve cells, do not divide because they are arrested permanently in the G0 phase. Experiments have shown that many other cell types have only a limited capability to divide. Normal cells grown outside of the body divide only a certain number of times and then stop. These observations suggest that cessation of mitosis is a normal, genetically programmed event. According to this view, aging genes are part of the genetic blueprint at birth. These genes have an important function in normal cells, but their activity is slow over time. They bring about aging by slowing down or halting processes vital to life. Another aspect of aging involves telomeres, specific DNA sequences found only at the top of each chromosome. These pieces of DNA protect the tip of the chromosomes from erosion and from sticking to one another. However, in most normal body cells, each cycle of cell division shortens the telomeres. Eventually, after many cycles of cell division, the telomeres can be completely gone, and even some of the functional chromosomal material may be lost. These observations suggest that erosion of DNA from the tip of our chromosomes contribute greatly to the aging and death of cells. It has recently been learned that individuals who experience high levels of stress have significantly shorter telomere lengths. Glucose, the most abundant sugar in the body, plays a role in the aging process. It is haphazardly added to proteins inside and outside cells, forming irreversible cross-links between adjacent protein molecules. With advancing age, more cross-links form, which contributes to the stiffening and loss of elasticity that occur in the aging tissues. Free radicals produce oxidative damage in lipids, proteins, or nucleic acids. Once again, free radicals produce oxidative damage in lipids, proteins, or I'm doing it again. Sorry. Free radicals produce oxidative damage in lipids, proteins, or nucleic acids by stealing an electron to accompany their unpaired electrons. Some effects are wrinkled skin, stiff joints, and hardened arteries. Normal metabolism, for example, aerobic cellular respiration in mitochondria, produces some free radicals. Others are present in air pollution, radiation, and certain foods we eat. Naturally occurring enzymes in peroxisomes and in systole normally dispose of free radicals. Certain dietary substances, such as vitamin E, vitamin C, beta-carotene, zinc, and selenium, are antioxidants that inhibit free radical formation. Some theories of aging explain the process at the cellular level, while others concentrate on regulatory mechanisms operating within the entire organism. For example, the immune system may start to attack the body's own cells. The autoimmune response might be caused by changes in cell identity markers at the surface of the cells that cause antibodies to attach to the cell and mark them for destruction. As changes in proteins on the plasma membrane of cells increase, the autoimmune response intensifies, producing the well-known signs of aging. In the chapters that follow, we will discuss the effects of aging on each body system in sections similar to this one. Now, I stumbled through that a little bit, and you might notice I've been stumbling at some points, and other points I just ramble right through it. But I want to repeat that one again, the free radical portion, because I think it's important. And observations can be made here, and they might apply to you directly. Free radicals produce oxidative damage in lipids, proteins, or nucleic acids by stealing an electron to accompany their unpaired electrons. Some effects are wrinkled skin, stiff joints, and hardened arteries. Normal metabolism, for example, aerobic cellular respiration in mitochondria, produces some free radicals. Others are present in air pollution, radiation, and certain foods we eat. Naturally occurring enzymes in peroxisomes and in cytosol normally dispose of free radicals. Certain dietary substances, such as vitamin E, vitamin C, beta-carotene, zinc, and selenium, are antioxidants that inhibit free radical formation. Some theories of aging explain the process at the cellular level, while others concentrate on regulatory mechanisms operating within the entire organism. Here's the point where I wanted to continue. For example, the immune system may start to attack the body's own cells. This autoimmune response might be caused by changes in cell identity markers at the surface of cells that cause antibodies to attach to and mark the cell for destruction. As changes in the proteins on the plasma membrane of cells increase, the autoimmune response intensifies, producing the well-known signs of aging. So, as a lot of you, some of you already know, autoimmune disease may be caused by environmental variables that exist. Whether they be toxic chemicals, perhaps free radicals, which would be an environmental toxin, that cause a change in the outer cellular membrane, which could mark it for death or damage the cell in some way. Because we know that constant contact to a toxin can cause, and I should rephrase that, constant contact to a substance, even if it is not a known toxin like latex. Some people are allergic to latex, some people are not. However, some people who experience continual contact with latex develop a sensitivity towards it. Therefore, latex affects the cells in such a way that you become allergic to it. It has affected your autoimmune system. It certainly is worth thinking about. So the clinical connection: progeria and Werner syndrome. Progeria is a disease characterized by normal development in the first year of life, followed by rapid aging. It is caused by a genetic defect in which telomeres are considerably shorter than normal. Symptoms include dry and wrinkled skin, total baldness, and bird-like facial features. Death usually occurs around age 13. Werner syndrome is a rare inherited disease that causes a rapid acceleration of aging, usually while the person is only in his or her 20s. It is characterized by wrinkling of the skin, graying of the hair, and baldness, cataracts, muscular atrophy, and a tendency to develop diabetes mellitus. Cancer and cardiovascular disease. Most affected individuals die before the age of 50. Recently, the gene that causes Werner syndrome has been identified. Researchers hope to use the information to gain insight into the mechanism of aging, as well as to help those suffering from the disorder. Now, beginning with this chapter, there is a section called Disorders, Homeostatic Imbalances. So I'm going to read that, and then I'm going to separate the study outline. And I'm going to label that so that for quick reference you can go back and just have that. So, disorders, homeostatic imbalances. Most chapters in the text are followed by concise discussions of major diseases and disorders that illustrate departures from normal homeostasis. They provide answers to many questions that you might ask about medical problems. Cancer. Cancer is a group of diseases characterized by uncontrolled or abnormal cell proliferation. When cells in part of the body divide without control, the excess tissue that develops is called a tumor or neoplasm. The study of tumors is called oncology. Tumors may be cancerous and often fatal, or they may be harmless. A cancerous neoplasm is called a malignant tumor or a malignancy. One property of most malignant tumors is their ability to undergo metastasis, the spread of cancerous cells to other parts of the body. A benign tumor is a neoplasm that does not metastasize. An example is a wart. Most benign tumors may be removed surgically if they interfere with normal body function or become disfiguring. Some benign tumors can be inoperable and perhaps fatal. Types of cancers. The name of a cancer is derived from the type of tissue in which it develops. Most human cancers are carcinomas, malignant tumors that arise from epithelial cells. Melanomas, for example, are cancerous growths of melanocytes, skin epithelial cells that produce the pigment melanin. Sarcoma is generally a term for any cancer arising from muscle tissue or connective tissue. For example, osteogenic sarcoma. The most frequent type of childhood cancer destroys normal blood tissues. It destroys normal bone tissues. Leukemia. Now I'm stumbling a little bit here, and I'm going to go back and tell you why in a moment. But leukemia is a cancer of blood-forming organs characterized by rapid growth of abnormal leukocytes. Lymphoma is a malignant disease of lymphatic tissue. For example, a lymph node. Now, the reason I'm stumbling is because of an interesting fact here. Carcinoma, from the root word carcin, which means cancer, and omus, which means tumor. So carcin, as in carcinogen, cancer. Melanoma, well, that means black. And that makes total sense because a melanoma is a cancer of the pigment producing cells. Melan, black. Melan, M-E-L-A-N, melanoma. Sarcoma, sarc equals flesh, is a general term for any cancer arising from muscle tissue, muscle cells, or connective tissue. Osteo, as in osteogenic sarcoma, osteo meaning bone. And that's the most frequent type of childhood cancer that destroys normal bone tissue. Leukemia, leuk, L-E-U-K, meaning white, emia, meaning blood, white blood, is a cancer of blood-forming organs characterized by rapid growth of abnormal leukocytes, which are white blood cells. So I'm going to read that again. Leukemia, leuk, meaning white, emia, meaning blood, is a cancer of blood-forming organs characterized by rapid growth of abnormal leukocytes, white blood cells. Lymphoma is a malignant disease of lymphatic tissue, for example, of the lymph nodes. Growth and spread of cancer. Cells of malignant tumors duplicate rapidly and continuously. As malignant cells invade surrounding tissues, they often trigger angiogenesis, the growth of a new network of blood vessels. Proteins that stimulate angiogenesis in tumors are called tumor angiogenesis factors, or TAFs. The formation of new blood vessels can occur either by overproduction of TAFs or by the lack of naturally occurring angiogenesis inhibitors. As the cancer grows, it begins to compete with normal tissue for space and nutrients. Eventually, the normal tissue decreases in size and dies. Some malignant cells may detach from the initial tumor and invade a body cavity or enter the blood or lymph that circulate and invade other body tissues, establishing secondary tumors, metastasis. Malignant cells resist the anti-tumor defenses of the body. The pain associated with cancer develops when the tumor presses on nerves or blocks a passageway in an organ so that the secretions build up pressure, or as a result of dying tissue or organs. Causes of cancer. Several factors may trigger a normal cell to lose control and become cancerous. One cause is environmental agents, substances in the air we breathe, the water we drink, and the food we eat. A chemical agent or radiation that produces cancer is called a carcinogen. Carcinogens induce mutations, permanent changes in the DNA-based sequence of a gene. The World Health Organization estimates that carcinogens are associated with 60 to 90% of all human cancers. Examples of carcinogens are hydrocarbons found in cigarette tar, radon gas from the earth, and ultraviolet radiation in sunlight. Intense research efforts are now directed towards studying cancer-causing agents or oncogenesis. Let me correct that. Oncogenes. Intensive research efforts are now directed towards studying cancer-causing genes, oncogenes. When inappropriately activated, these genes have the ability to transform a normal cell into a cancerous cell. Most oncogenes derive from normal genes called proto-oncogenes. They regulate growth and development. The proto-oncogene undergoes some changes that cause it either to be expressed inappropriately or to make its products in excess amounts or at the wrong time. Some oncogenes cause excessive production of growth factors, chemicals that stimulate cell growth. Others may trigger changes in a cell surface receptor, causing it to send signals as though it were being activated by a growth factor, which is very similar to what I was explaining about immune diseases. Others may trigger changes in a cell surface receptor, causing it to send signals as though it were being activated by a growth factor. As a result, the growth pattern of the cell becomes abnormal. Proto-oncogenes in every cell carry out normal cellular functions until a malignant change occurs. It appears that some proto-oncogenes are activated to oncogenes by mutations in which the DNA of the proto-oncogene is altered. Other proto-oncogenes are activated by a rearrangement of the chromosomes so that segments of the DNA are exchanged. Rearrangement activates proto-oncogenes by placing them near genes that enhance their activity. Some cancers have a viral origin. Viruses are tiny packages of nucleic acids, either RNA or DNA, that can reproduce only while inside the cells they infect. Some viruses, termed oncogenic viruses, cause cancer by stimulating abnormal proliferation of cells. For instance, HPV, the human papillomavirus, causes virtually all cervical cancers in women. The virus produces a protein that causes proteasomes to destroy p53, a protein that normally suppresses unregulated cell division. In the absence of this suppressor protein, cells proliferate without control. Now, it's very interesting that they would say HPV, human papillomavirus, and then they would mention only women. Because in some literature they say human papillomavirus, which does not have symptoms in a male. Well, it may be that through the scientific method, this book is changed in the future because we know that males have prostate cancer. And it may very well be that one way to establish prostate cancer in a male is through the HPV. It may be. We don't know that. Human papillomavirus may be one of the root causes of prostate cancer. We also can theorize that an STD might mutate a cell and cause prostate cancer in a male. So you don't know what you're getting into, so to speak. You might find out later on. It's just a theory at this point. It's out there a lot. Anyway, be careful with that. And so, recent studies suggest that certain cancers may be linked to a cell having an abnormal number of chromosomes. As a result, the cell could potentially have extra copies of oncogenes or too few copies of tumor suppressor genes, which in either case could lead to uncontrolled cell proliferation. There's also some evidence suggesting that cancer may be caused by normal stem cells that develop into cancerous stem cells capable of forming malignant tumors. Later in the book, we will discuss the process of inflammation, which is a defense response to tissue damage. It appears that inflammation contributes to various steps in the development of cancers. Some evidence suggests that chronic inflammation stimulates the proliferation of mutated cells and enhances their survival, promotes angiogenesis, and contributes to invasion and metastasis of cancer cells. There is a clear relationship between certain chronic inflammatory conditions and the transformation of inflamed tissue into malignant tissue. For example, chronic gastritis, inflammation of the stomach lining, and peptic ulcers may be a causative factor in 60 to 90% of stomach cancers. Chronic hepatitis, inflammation of the liver, and cirrhosis of the liver are believed to be responsible for about 80% of liver cancers. Colorectal cancer is 10 times more likely to occur in patients with chronic inflammation. Chronic inflammatory diseases of the colon, such as ulcerative colitis and Crohn's disease, and the relationship between asbestosis and silicosis, two lung inflammatory conditions. That is the beginning of a new sentence. And the relationship between asbestosis and silicosis, two chronic lung inflammatory conditions in lung cancer, has long been recognized. Chronic inflammation is also an underlying contributor to rheumatoid arthritis, Alzheimer's disease, depression, schizophrenia, cardiovascular disease, and diabetes. That's very important, because we are hearing about rheumatoid arthritis more and more in the news with the passing of Glenn Fry and the biologic response modifier drugs, which in some instances inhibit proliferation of tumor necrosis factor alpha, which lead to the tumor necrosis factor alpha cell is a killer cell that attacks tumors. So on one hand you have a drug depleting that, and you could say, oh, the cancer came from the depleting of TNFA, and on the other hand you can say the person had rheumatoid arthritis, and so their inflammatory process caused the cancer. So it's very thin line here, but it's very interesting to know that chronic inflammation is an underlying contributor to rheumatoid arthritis, to Alzheimer's disease. How often do you hear that a contributing factor to Alzheimer's disease is chronic inflammation? I don't know. I don't think a lot. Did you know that chronic inflammation can lead to depression, schizophrenia? Cardiovascular disease, we could surmise that. Chronic inflammation leads to diabetes. That's new information to me. So, very interesting. Carcinogenesis, a multi-step process. Carcinogenesis, the process by which cancer develops, is a multi-step process in which as many as 10 distinct mutations may have to accumulate in a cell before it becomes cancerous. The progression of genetic changes leading to cancer is best understood for colorectal cancer. Such cancers, as well as lung and breast cancer, take years or decades to develop. In colon cancer, the tumor begins as an area of increased cell proliferation that results from one mutation. This growth then progresses to abnormal, but non-cancerous growths called adenomas. After 2 or 3 additional mutations, a mutation of the tumor suppressor gene p53 occurs and a carcinoma develops. The fact that so many mutations are needed for a cancer to develop indicates that cell growth is normally controlled with many sets of checks and balances. A compromised immune system is also a significant component in carcinogenesis. In automotive, I would call that a series of misfortunate events. You bring the vehicle in. It has a broken fan belt. Well, okay, you might replace that fan belt, but then it breaks again. Well, okay, wait a second now. The series of misfortunate events would be the water pump froze. When the water pump froze, it broke the belt. When the water pump stopped pumping coolant, It warped the head, blew the gasket, cracked the head. So you have a multi-step process, a series of unfortunate events that caused the demise of the engine. And this is why I continue to say that mechanics is mechanics. And that's basically what we're talking about here. All the terms are different. A lot of the terms are different. But the theories are the same. Engineering is engineering. And so, treatment of cancer. Many cancers are removed surgically. However, when cancer is widely distributed throughout the body or exists in organs such as the brain whose function would be greatly harmed by surgery, chemotherapy and radiation therapy may be used instead. Sometimes surgery, chemotherapy, and radiation therapy are used in combination. Chemotherapy involves administering drugs that cause death of cancerous cells. Radiation therapy breaks chromosomes, thus blocking cell division. Because cancerous cells divide rapidly, they are more vulnerable to the destruction effects of chemotherapy. Because cancerous cells divide rapidly, they are more vulnerable to the destructive effects of chemotherapy and radiation therapy than a normal cell. Unfortunately for patients, hair follicle cells, red bone marrow cells, and cells lining the gastrointestinal tract are also rapidly dividing. Hence, the side effects of chemotherapy and radiation therapy include hair loss due to death of hair follicle cells, vomiting and nausea due to death of cells lining the stomach and intestines, and susceptibility to infection due to slowed production of white blood cells and red bone marrow. Treating cancer is difficult because it is not a single disease and because the cells in a single tumor population rarely behave all in the same way. Although most cancers are thought to derive from a single abnormal cell, by the time a tumor reaches a clinically detectable size, it may contain a diverse population of abnormal cells. For example, some cancerous cells metastasize readily and others do not. Some are sensitive to chemotherapy and some are drug resistant. Because the difference in drug resistance, a single chemotherapeutic agent may destroy susceptible cells but permit resistant cells to proliferate. Another potential treatment for cancer is currently under development in virotherapy, the use of viruses to kill cancerous cells. The viruses employed in this strategy are designed so that they significantly target cells without affecting the healthy cells of the body. For example, proteins such as antibodies that specifically bind to receptors found only in cancer cells are attached to viruses. Once inside the body, the virus binds to cancer cells and then infects them. The cancer cells are eventually killed once the viruses cause cellular lysis. Researchers are also investigating the role of metastasis regulatory genes that control the ability of cancer cells to undergo metastasis. Scientists hope to develop therapeutic drugs that can manipulate the genes and therefore block metastasis of cancer cells. Medical terminology. Most chapters in this text are followed by a glossary of key medical terms that include both normal and pathological conditions. You should familiarize yourself with these terms because they will play an essential role in your medical vocabulary. Some of these conditions, as well as ones discussed in the text, are referred to as local or systemic. A local disease is one that affects one part of a limited area of the body. A systemic disease affects the entire body or several parts. The science that deals with why, when, and where diseases occur and how they are transmitted in a human community is known as epidemiology. The science that deals with the effects and uses of drugs in the treatment of disease is called pharmacology. Anaplasia, the loss of tissue differentiation and function that is characteristic of most malignancies. Atrophy, a decrease in the size of cells with a subsequent decrease in the size of the affected tissue or organ wasting away. Dysplasia, alteration in the size, shape, and organization of cells due to chronic irritation or inflammation. May progress to neoplasia, tumor formation, usually malignant, or revert to normal if the irritation is removed. Hyperplasia, increase in the number of cells of a tissue due to an increase in the frequency of cell division. Hypertrophy, increase in the size of cells without cell division. Metaplasia, the transformation of one type of cell into another. progeny, offspring, or descendants. Proteomics, the study of the proteomere. Proteomics, the study of the proteome. In order to identify all the proteins produced, it involves determining the three-dimensional structure of proteins so that drugs can be designed to alter protein activity to help in the treatment and diagnosis of disease. Tumor marker, a substance introduced into circulation by tumor cells that indicate the presence of a tumor as well as specific type. Tumor markers may be used to diagnose, make a prognosis, evaluate a response to treatment, and monitor for recurrence of cancers. Okay, I'm going to take a break. I'm going to come back and I'm going to do the study outline. Which is a summary of what we've studied so far.